In this lesson, I'll briefly review the operation of neurons and then share a relatively new technique that scientists have developed to visualize neuron activity. And it involves genetically engineering these neurons to emit light. We've learned about the three basic parts of the neuron: the dendrites, the branches here, the cell body, and then the axon and terminal. The dendrites are the receiving zone of the neuron, so these branches provide lots of space for hundreds or thousands of synapses from other neurons in a brain. At each synapse site, if that synapse is active, in other words, electrical signal has made it down to the terminal and neurotransmitters are released, that generates a small electrical signal at the dendrite at that location. If there are lots of active synapses at the same time, the cell body is going to sort of detect that, and if it passes a threshold amount of activity on the dendrites out here, the cell body will generate an output signal, and that electrical signal races down the axon. This is an all or nothing signal. But the generation of this signal reflects the fact that there was lots of activity out here on the synapses around the dendrites. If there is not much activity here, the neuron will stay quiet. It will not generate an output signal. When the electrical signal races down and gets to the terminal, neurotransmitters are released, and these will generate an electrical signal in whatever target cell is at that location. Let's take a look at an animation that captures this process. Now, this is a visual animation. So here we see the structure of a neuron here, much like the diagram down here. We've got these little red stars that are being produced and then moving down towards the cell bodies. They would be our little electrical signals here. Now, they're having them move down the dendrites to the cell body to sort of capture the idea that the cell body is adding up all of this electrical activity. It metaphorically has to make a decision. Should it generate an electrical signal or not? So evidently there is enough activity at synapses around here, there were enough little electrical signals being generated here that it added up to pass a certain threshold, and the cell body is generating an electrical signal as an output. That's the yellow flash that races down the axon and goes to the terminals. At the terminals here, neurotransmitters would be released to activate the target cells. So you're seeing they're trying to sort of visualize what neurons are doing, but electrical signals are not visible. They don't emit visible light. We wouldn't see this. Actually, we wouldn't see anything really happening if we were looking at a regular neuron in a brain. However, neurons can be genetically engineered so that they do flash light. So they will fluoresce when the neuron generates an electrical signal. So in a sense, genetically engineered neurons can be visually seen, kind of like what this animation is showing us right now. Now, how do they do that? We mentioned genetic engineering. There are other species of animals on planet Earth. Some of them are fluorescent. They fluoresce, and they have DNA that makes a protein, or that codes for a protein that allows the creature to fluoresce. So scientists have inserted some of these foreign genes into neurons, so the neuron will make these proteins that then fluoresce when the neuron generates an electrical signal. So they had to do a bit of engineering and to kind of put some genes together to engineer some proteins that will fluoresce only when the neuron generates an electrical signal. And so that then became a system where you could visually see neurons as they become activated. When the neuron generates an electrical signal, just like a typical neuron, it's also emitting light, and so we'll be able to visualize the activity of neurons. Okay, so this is going to be a video then that will show us the activity of a population of neurons in the cortex of an animal. You're seeing the surface of the brain here and some big blood vessels here. And you can already kind of see some little white specks here. These are going to be neurons that are going to be flashing on and off as they are responding to their inputs. So let's go ahead and play the video. So what you're witnessing now, so this is a microscope that is set up right to see the surface of the brain of this animal. You're witnessing neurons flashing according to their activity. So when a neuron is flashing, it means that particular neuron reached its threshold and is generating an electrical output signal. So we're able to visualize the activity of neurons because they've been genetically engineered to emit light whenever they generate an electrical signal. Now, you'll recall from our map of the human cerebral cortex, we've got different regions of the brain that are processing different kinds of information. So let's imagine this scene here was visual cortex, right? Well, then what you're witnessing as we play this video, what you're witnessing then, this would be what the brain is doing when we are seeing something. It's a population of a certain set of neurons are responding to whatever it is that we were seeing. And if we were seeing something different, it would be a different set of neurons that get turned on by that stimulus in the environment. So this is just it's a fascinating way to kind of think about what's happening in our brains. We can't feel the electrical activity of individual brain cells. There are, millions and millions, actually hundreds of millions, billions. The cortex might have, 18, 20 billion neurons. We can't feel the electrical activity of individual neurons. But here we're taking sort of we're stepping outside of a brain and looking at brain activity from the outside. Of course, if this were visual cortex of an animal, the animal would be having a visual experience of some kind. Let's take a look at a more magnified view. So here we have the section of cortex, and now we're going higher magnification. Again, we'll see these big tubes here. These are blood vessels, and you'll actually see blood running through the blood vessels. And now you can also see some of these white spots here. They're going to be flashing neurons. Let's take a look. So you can see the blood moving through blood vessels. And all these neurons have been genetically engineered to emit light whenever they produce an output electrical signal. So again, you're witnessing what brains are doing. Brains are made up of populations of neurons that are hooked up in complicated ways, but those networks are processing information. If again this were visual cortex, this is what it is to see something, but from an outsider's perspective. If this was our brain and we were looking at a sunset, this is what our brain is doing, and we are having the experience of a sunset. That, by the way, is a profound puzzle to modern science. How is it that the activity of a population of neurons here is what it is to see a sunset, right? It's a very, very deep puzzle. And scientists and philosophers have been working on that for hundreds of years. Now, one more example of this can be seen in this situation here. In the top right here, we have a little fish in a small bowl of water, and you're going to see a little white speck swimming around. That's a paramecium, and that would be food to the fish. Now, the fish is anchored, so it's not going to be able to swim after its food, but it's going to be watching. Down here, we're going to witness the eyes of the fish moving around as it tracks its prey, swimming around in the fishbowl. Over here, again, we see the surface of the fish's brain. And again, these neurons have been engineered to express a protein that will fluoresce when the neuron is activated. So we're going to be seeing in real time the pattern of brain activity that is going on as the fish is tracking its prey. Let's take a look. All right, so here we are. So in the top right here, we have the anchored fish, and we have the paramecium swimming around. Down here, we see the fish's eyes are tracking its moving prey. And over on the left here, we see the surface of the fish's brain here, and the little blue-green flashes are activated neurons. So you're seeing in real time the pattern of brain activity as the fish is tracking its prey. And again, it's profoundly puzzling that we are witnessing the fish's brain from the outside. So we're seeing what the fish's brain is doing. But from the fish's point of view, it's seeing, it's having an experience, a visual experience of its prey swimming around in this bowl. And that is the profound puzzle. How is it that the activity of neurons, the electrical activity of neurons, is somehow the same thing as having a conscious experience of something?